All right, everyone. How are you doing? We are kicking out the semester here. You'll have this assignment and your final. So, by all means, we're almost there. Today, we're going to be talking about species distribution modeling. This is both the lecture and the coding guide. There's going to be a lot of code in here that you'll need to do for the modeling, but I'm not going to cover in depth because you can spend way more than a week on this topic, more than a week understanding all the code and what it means. You'll get the basics down, understand the flow and the structure of the coding. You'll be able to produce the modeling metrics. And you'll be able to produce a species distribution model in the state of Colorado, at least. So we are going to be modeling a species in Colorado. I will provide you with the Colorado shapefile, as well as the predictor variables needed to produce a species distribution model. You will be using the SPOCC package, which stands for species occurrence, or something like that, and the OCC function to download species occurrence data, and basically you'll be switching out the occurrence data that I provide for this lecture guide and for species that you have that occurs within the state of Colorado. There are certain restrictions on what you need as for a data set, for an occurrence data set. That'll be provided in the assignment instructions. So let's not waste any time and let's just dive right in. So, species distribution modeling, like I said, is a dense topic. There are so many different algorithms to use and methods for model preparation. You can spend an entire career researching SDM, which people do. But here we're going to be covering species distribution modeling flying high at 30,000 feet. You'll be able to define species distribution modeling, understand its importance in ecological and social studies, and be able to recreate a generalized linear model from this tutorial. So species distribution modeling is the process of using species occurrence data, the observed XY location of an organism, and predictor variables, climate, soils, topography, land use, any type of continuous raster data to predict the spatial distribution of a given species. It's also known as ecological niche modeling, habitat suitability modeling, and range mapping. SDM is a common tool used in conservation biology and bioregional planning. So, a little image that kind of shows what we're going to be doing. So these dots, let's just say they are species occurrences within this domain here, this gray box. Using an algorithm to analyze the occurrence data with environmental predictors, so terrain, climate, soils, land cover, you can produce the predicted suitability of a given species. So here's a couple of things to look at as you're building these. Species distribution modeling does not provide information on how many species are present in a location, which would be the abundance, or if a species is actually even there. So, let's say that this is the predicted distribution of a species, predicted habitat suitability from low being in yellow to high of the dark purple here, or low being here and high being here on the bar. That is not saying that there are more species present at that moment in the higher suitability, or actually saying that a species is technically present. Species distribution modeling outputs are probability representations of habitat that is suitable for a given species. So, the higher the suitability doesn't mean that there are more species currently occupying that area or that they are even there, but based on the conditions, like in this model, terrain, climate, soil, and land cover, these areas are higher suitability for a species to be in. So we don't know the number of how many are there, we don't know if it's actually there, but based on the model of occurrence data and predictor variables in terrain, climate, soils, land cover, what have you, that is an area of higher suitability, so a species could persist within that area. Species distribution modeling is an effort to define a suitable habitat. The goal of the model is to represent reality. However, all models, statistical models, spatial models, they're inherently wrong. All models are wrong. They can be useful and powerful tools in ecology, but it's important to understand that species distribution modeling has limitations. So what does that mean, wrong? It's wrong because it doesn't fully represent reality, because we, as humans, do not have the capability to fully represent reality. We're using models to show, based on the predictors that we include and the data that we include, to represent reality. So there's no model that is 100% true in species distribution modeling, but they could produce a model showing high predicted suitability where there are areas of high predicted suitability. They have their uses, and that doesn't mean that they're bad. Species distribution modeling is used in a number of, conservation, ecology, bioregional planning, human environment systems projects. Feel free to go on Google Scholar and type in species distribution modeling. Very common tool, and it has great use into understanding the distribution of species and identifying habitat. But if you produce a model, you cannot definitively say this model is true. So we're going to be using generalized linear models. A generalized linear model can be applied in univariate and multivariate applications, and it is used to estimate an ecological response as a linear combination of independent predictor variables. So basically this is saying, in a sense, it acts as a logistic regression. There are certain thresholds between predictor variables that would either represent the suitable habitat of a species and the non-suitable habitat of a species. So, for example, we could be looking at the suitability between 0 and 1, 1 being highly suitable, 0 being not suitable at all. Now, based upon a predictor variable x, let's just say it's elevation. Based upon, as we change in elevation, we could go from non-suitable to highly suitable and somewhere in between. A generalized linear model will do this between many predictor variables in the response of an occurrence data of occurrence variable like our occurrences and the predictor variables. So it just generalized linear models act as a logistic regression. Model requirements, SDM require predictor variables x and a response variable y. So response variable y, predictor variable x. Predictor variables are explanatory variables based on occurrence location. The response variable, which we'll talk about in a little second here. The predictor variables help explain and produce a predicted distribution of a species of interest. For example, let's say there is a very green plant species that occurs on northern slopes, steep slopes, and at elevations higher than 500 meters. This is my terrible figure drawn in paint. And let's say that this is the species. So, they occur on northern slopes. This is north, this is south. They occur on steep slopes, so very steep slopes compared to other slopes that are less steep, and at elevations higher than 500 meters. So, based on the specific values of each occurrence in elevation, slope, and aspect, we can get a good idea of the geographic range of habitat suitability for that very green plant species. So, if someone just gave you this little diagram without the green and said, okay, fill in an area that is above 500 meters on the steepest slopes and on north sides, you'd probably shade in right here. So that is species distribution modeling in a sense. Many, studies people, they define the distribution of a species or a suitable habitat like this. It's very simple. And you can kind of do that qualitatively. An algorithm like GLM, so Generalized Linear Model, allows us to include many variables and examine their influence on habitat suitability computationally. So less qualitative, more quantitative. In fact, entirely quantitative. It allows us to see what we may not see in the difference between highly suitable and low suitable habitat between either values within a predictor variable or across predictor variables. So while this may be a way to identify suitable habitat, and in a way it is species distribution modeling, we use statistical algorithms or classifiers, and these can range from parametric, semi-parametric, non-parametric in scope to produce the spatial distribution of a species. So there's a lot of different algorithms, and they have their certain biases, and this is just one, and it is very commonly applied in species distribution modeling. The response variable is the species occurrence. The response variable guides the selection of variable importance in the distribution of a given species by spatially overlaying the occurrence points of a species on the predictor variables. We can see which values are associated with each occurrence point, which this will make more sense in a second, but ideally you're understanding the values, so of these little points here, the values of each predictor variable that is associated with each point. And then given the model, which we see that defines between highly suitable and low suitable habitat, we can produce a model. So, what are we going to be using? This example is of the Colorado state tree, the blue spruce. Our specific species of interest for this example will be the state tree of Colorado, like I just said, the blue spruce. Blue spruce is from the spruce family, Picea, and its genus pungens, what have you, is Latin for sharp, biting, or piercing. If you ever come in contact with a blue spruce, you can understand why it is named pungens. It's sharp, biting, and piercing. They're very sharp needles. Blue spruce is generally green, but can look very blue due to a wax that reflects blue light on its needles. Going back to your remote sensing stuff. Blue spruce can be found in the southern Rockies at 6,000 to 10,000 feet. But it is also planted in trees, yards. You'll find it at much lower elevations in urban areas. For example, I have a small blue spruce. Man, that's going to be fun. In my front yard, and we're at about 4,600 feet, and it's doing just fine. They also make an excellent Christmas tree. So there you go. Gathering species data via the SPOCC package, and so I'm posting another video on how to do that. I will walk you through the code here quickly because I will also have the code provided in a R script and the video. But you choose the SPC list for where we're doing Picea pungens. I don't know, I'm just going to say pungens from now on. So you are using the genus and species to run the SPOCC within the SPOCC package and the OCC function to query that species from a list of databases that store the occurrence data of species. And so I am not going to cover this because there is another video and another script, but we take the outputs and we turn that into a CSV, which you may save. And what I have done here is just save that as CSV and loaded it. So after you collect the species data, which will be done in that other video, you can load it in and give it a name. So Picea pungens, I forget which one I said I was going to use. I just called it PIPU, this 4-letter code, and then OCC raw. And I'm loading in that CSV right here, and I'm just using a pandas to look at it. So the output data set, by using the OCC function, you will have a numbered list. You will have the species name. You will have longitude and latitude. You will have the provider. In this case, it is GBIF, which is Global Biodiversity Informatics something. You will have the date that it was acquired, and you'll have a key unique to that given species. So by far the most important data that we will gather here is the longitude and latitude, or the X and Y coordinates of each occurrence. Perfect. So now that we have gathered our species occurrence data, we can load in our predictors. Now, the assignment is going to be entirely in Colorado because I am providing that information for you. It's a rectangular state, and its boundaries are defined quite easily. And yeah, by far the best for you because I am providing this information. So I'm providing you predictor variables, like we mentioned before, continuous raster data that describes the environmental characteristics that may drive the distribution of that species. So I'm providing to you elevation, slope, aspect. Now I'm also providing some some edaphic variables, so soils, so pH minimum, pH average, pH max, fragments 3 to 10. You get into your sieves, dry weight, OMR, why am I forgetting? Organic matter something. KSAT, so K saturation, average water content, water saturation. And so you don't have to look for these. If you were doing a modeling study in a different area at a different extent, you would have to acquire your own predictor variables and create your own raster stack that contains all of these. There are also, after the this just shows the first 16 variables within this stack. You have water saturation. So after water saturation, I've also included sand, silt, clay, and 19 bioclimatic variables. And these variables are very common. So 19 bioclim variables, I encourage you to Google them and look at them. But you'll see things like mean temperature average, mean precipitation average. So a lot of the climatic drivers of vegetation. So this, what we're doing this study for is of the blue spruce, so a plant. Some of these variables may not be important if you're modeling a agile species. So like let's say you're modeling the distribution of pumas. So puma concolor, the American mountain lion. Soil conditions may not be very useful in modeling that species, but elevation, slope, aspect, and climate definitely would be. You could also add in land cover as highly urbanized areas probably aren't a great suitability indicator of pumas. But then you could also look into roughness, like we calculated earlier in the semester. Roughness would be how rough the terrain is given the span of the cell. That'd be a very good indicator of mountain lions. So for plants, definitely soils, topography, and climate are the big drivers of distributions. That's what we're using for this study. So I have loaded in my species using the raster or the rast function from Terra and plotted them. And here I'm just plotting the first one, a 1 kilometer elevation of Colorado. So all of the predictors here, they must be the same spatial resolution in any of your studies for species distribution modeling. You can't have, elevation be 30 meters while your climate is at 4 kilometers. They must all be the same spatial resolution. So all of the predictors within this study are at 1 kilometer spatial resolution, which means the cell for each raster is 1 kilometer by 1 kilometer. Great. And if you're familiar with Colorado, this is the Southern Rockies. You have the Colorado Front Range, the San Juans. Over here you have Rocky Mountain National Park. You get down to the Sangre de Cristos. And then over here you have the mass that goes into the Great Plains. And so right around here you'll have Fort Collins, Boulder, Denver, and then you can drop down into Colorado Springs, stuff like that. And many ski towns in between. If you're not familiar with Colorado, please look at a map. It'll help you understand the final output of the species distribution model. That will help you understand, did my model actually work? Does it look right? But let's do some organization and develop a few maps along the way. So a lot of this stuff we're going to cover quickly because the code gets very in-depth very quick, and I don't want you wasting your time trying to understand the specifics of every single line of code we are building here when you could produce your own species distribution modeling outputs and take time later to understand if you're more interested. And if you are more interested, you could even do this for a final project. I have the code for several different algorithms, machine learning algorithms and boosted regression trees, random forest, a semi-parametric model of generalized additive models, maximum entropy, which is a very commonly used algorithm in species distribution modeling. It is also known as maxent. And then a Bayesian model of Bayesian additive regression trees. So if you are interested in doing that for a final project, you could. But right now we are going to be organizing our data, creating pseudo-absences, and seeing what we got. So I'm calling this spec raw, and from my raw occurrence data that we collected right here from the OCC function, I'm getting rid of all null values, and I'm setting this as st as sf to create an sf object. I'm using my coordinates of longitude and latitude. It goes x then y, no matter if you're using meters or what have you. Then I'm assigning a coordinate system, 4326, which is world, I think it's WGS 84, which uses latitude and longitude. So I've created an SF object based upon that raw data, gave it a CRS. You have done this many times so far, so this should look familiar. I'm reading in the state of Colorado, and well, it's actually the states, which I'll provide, so it's in any of the states, but we're going to subset this to Colorado. We're filtering it down using the dplyr package, and using the percentage greater than percentage sign. You have also done this many times to subset data. And then we are going to crop all of the occurrences to the state of Colorado. So when you collect species occurrence data via OCC function, you can restrain where you are selecting the data or where you're gathering the data from. So you can add in a box. It's easiest just to download it, which will download probably for all of North America, and up it to whatever your modeling domain is. Looking at this should be its own separate title here, so you can ignore that. This just treat it as something big like this. But I have cropped the occurrence data and plotted them within Colorado. This rectangle is Colorado, and here we have occurrences of Colorado blue spruce. So, based on what I'm looking at here, you know you have the mountains, right? So you're going to find Colorado blue spruce somewhere here. Honestly, this is probably a good distribution model in itself. But you can see here, we probably have some planted in urban areas that were thrown into one of these open source occurrence data databases. There we go. We have our presences, and they are spatial, and they are cropped to Colorado. Now, we are going to be creating pseudo-absences, and so this is where this title comes in. Pseudo-absences. So species distribution modeling often requires the use of species absence points. Just as presence points tell us which values for each predictor variable are useful in modeling a species distribution, absence tells us which values are not important. So this will help us understand greatly our generalized linear model based upon absence and presence. So let's say you have a bunch of species presence in these values in 6 to 10, and you have a lot of absence points from 0 to 4, you can get a pretty good idea of where your suitability will lie based on a logistic regression. So that's why our absence points are extremely important, and this is not just for GLMs, this is for random forest, boosted regression trees, support vector machines, every other species distribution modeling algorithm. There are some that are presence only, but we're going to be looking at presence and absence, as absence does play a significant role in the shaping of distributions. So the use of absence allows us to define the overall habitat suitability of species, not just where it's predicted highly, but where it's not. We don't have actual species absences for this study. No one has gone out and kind of same thing but opposite of occurrence. So saying, oh, here's an area there is no blue spruce, I will mark its location. We don't have that information right now. So we're going to create them, and they're called pseudo-absences, or an X-fold pseudo-absence process. So since we do not have field-collected absence points, we are going to generate random points across our modeling domain, which is the state of Colorado. And since these points are random, we are going to be oversampling. So we're going to be doubling the amount of absence points for the presence points. And this means that, due to the off chance that an absence point is a presence point in reality, we are going to produce, double the amount of absence points to account for an uncertainty in our data set. So you can do this by creating, 3 times, 4 times greater the amount of presence points. But we are going to be doing just double the absence, double the absence of the presence. So I'm calling this PIPU dot abs, so for absence, and we're going to be using the function stsample. stsample allows us to develop. My wife just texted me randomly. Yellowstone is 150 years old. Okay. So we're going to be using the stsample function to generate random points. So within here, you define your boundary, and we're using stgeometry of Colorado. So we're the modeling domain of Colorado. The type we want to create is random. The size, and the size is not like, I want this point to be this big. It's how many points. So our size is going to be equal to the number of rows within our presence data set times 2. So the number of rows in our data set is how many presences we have, and we're going to be multiplying that by 2. So we have double the amount of absence points as we do presence points, and we are going to be using the st as st underscore sf to make sure that this is now becoming an SF object, which meaning it is a spatial object. So, that's simply put, we have developed a data set of absences that are double the amount of presences. And I skipped this, but it's set seed, and you put in numbers. Change this value for each species that you model when you're creating pseudo-absences. Set seed is a function to be able to have a reproducible sample. So, if I did this several times over, the absences would keep changing. But when I set the seed to 1, 1, 1, 1, that means every time I run this, it's going to be the same random numbers, the same random points, if that makes sense. But if you're doing a different species, you're going to want to choose this number. So this will be a reproducible way to make sure that we have the same random points. Now, plotting the Colorado geometry, plotting the absences in blue, and plotting the presence points in red, here we have it. So the whole modeling domain, we have a lot of absence points throughout, and even into areas where there could be presences, and that's why we doubled the amount, and then we have our red presence points. So there you go. Also, same thing as above. I don't know what happened to my R markdown. Wasn't paying close enough attention. We're going to be combining the presence and species absences. So, how we do this, we take abs chord, so the coordinates of the absence points, we're doing st coordinates. So instead of having the whole thing, we're just taking the coordinates, and now we are combining using the cbind function the absence coordinates with the presence coordinates. Are producing a column called PA, which stands for presence absence. If by chance, so if it is a absence points, we are assigning the presence absence column of 0, and the presence would be a 1. So you'll have the coordinates, and you'll have a column called PA. If it is a presence point, it will be a 1. If it is an absence, it will be a 0, and this will help the algorithm determine what is present and what is not. So we are doing pres chords. We're doing the coordinates now of all of them combined, or just the presences, and we are running ahead on the presence coordinates. So these are all presence points of x and y, and this is the head of our data that we have collected earlier and turned it into a simple feature. So this is the data from the OCC function. And now we are going to combine this data set with our coordinates, right? So now we have it here. We have the same data that we have collected in the OCC function, and then we have our x and y columns thrown in. Okay? Same thing for the absences. Taking a look at what we have created earlier, and then for absences we have the X, we have the Y, and we have PA, and these are all zeros because they are absent. And then we have the geometry because it is an SF feature. So we have created the PA column for the absences, but we have not created them for the presence points, so we are going to be adding in the presence absence column for the presence points. As we can see here, this is all the presence data. We do not have a PA column yet, but we will assign it a 1 because it is a presence, and we'll take a look at what we have created. Now, the absence data set only has X, Y, and PA, whereas our presence data set has X, Y, X.1, which is just the column number, name, provider, date, key, geometry, and then the PA. We don't need all of those, so we are going to get rid of most of them. So here is just a way to subset. PIPU is still going to be the same data set, but we are only taking 1 and 2, which is X and Y, and then we are taking 9, which is the PA, and then 8, which is the geometry column. So this is associated with each column. We are taking the first, second, 9th, and 8th column in that order, which will subset that to look much like our absence column. And as they are now the same in structure, we can combine them. So we are calling this now PIPU.PA, meaning dot presence absence, so Picea pungens dot presence absence. We are R binding, which is row binding, since they have the same number of columns. We are putting them together and we are creating a table. We're running the table function to see how many values of either absence or presence or absence that we have. So within our new created data set that contains the X, Y columns, as well as a PA column showing 1 for presence, 0 for absence, we have 5,504 absences and 2,752 presence points. Now, if you see, there are double the amount of absence points. So this is great. This is what we need. And if we take a look at now of our newly created data set, we will now see that we have X, Y, the presence absence column, and the geometry. We need this to train our data, and I'll tell you what that means right now. So training your data. Data training is needed for predictive modeling. Here we are simply extracting the values of each predictor variable to each species absence and presence point. So using the extract function, we are going to overlay all of our points onto each one of these 38 predictor variables and take out the value that occurs at that species presence or absence. So we have our predictors, right? We have our data set that we just created, the X, Y, PA, and the geometry. And Terra package, so here's what you need to be doing. So spec.pres.sv, that's what we're going to be calling it now. This will be turning our PIPU PA data set, right? This one right here, from an SF object to a SPAT vector. We must change it to a SPAT vector because we are working with SPAT rasters. So now we will be calling it spec.pres.extract. We will be using Terra, the package, and the function extract. First you put the predictors, so our predictor variables, and then we put our SPAT vector data set, so these presence and absence points right here. And this will extract all the values that this occurs at for each point. Now, we are going to be C binding, which is column bind from spec pres extract, what we have just done up here, to our presence and absence data set. So we'll be combining all the extracted values to this data set right here, and this is what it looks like. So this is the first 6 values in our newly created trained data set, where we have elevation, slope, aspect, and I'll show you that PHMN does not have values, so there's something wrong with this data, and I'll show you how to get rid of that. So for all of our predictor variables, the value that occurs at the spatial location of the presence or absence is now within this data set. So let's say for point number 1 here, it's at -106 longitude, 39 latitude. At that elevation, it looks like it is 3,044 meters. The slope is 5 degrees. The aspect is 126, which means that it is cold. It's about facing southeast, and then based on whatever these values are present. So, like for sand content, it looks like it's 53% sand content, 26% silt, 22% clay. So it basically just takes all the values for all the presence points. So there's a lot of values here, and we'll be using this once again based on presence and absence to perform a logistic regression to determine what is suitable and what is not. Okay, so we have created the data set. Here, we're just going to be using, I call it, you actually can ignore this. This is for the max n portion, which I did not provide here. But just looking at the names for our data, we have them right here. Perfect. We thus far gathered species occurrence data. We have organized them. We have created species absences. We have combined the species absence to the presence that were previously cropped to the state of Colorado. And now we have used all of those points to extract values of the predictor variables. Great. Now we are ready to model. And this is the section that we're going to be covering pretty quick, because I don't want you to waste your time trying to understand everything before we can even produce a model. Which ideally, you want to understand everything you're doing, but that's why we have statisticians. So we're going to be assigning the predictor variables, and we are going to be creating our data set, our modeling algorithm. Okay, so we're calling PREDS SPP PREDS. That's just the way my code was set up. And we're calling DAT1 our trained data. So this data set right here will become DAT1. I'm looking at the dimensions, the table, and the head. So here's the dimensions, here's the table, and then here's the head of the data, just the first two rows of the entire thing. So, in this data set, I have gotten rid of pH-min. And I have pretty much not shown you how to do it on purpose, but at the same time, you should know how to do it. So, looking at this data set, you will want to get rid of this pH-min column. So you will be doing something along the lines of this. So remember, in this right here, I said I have all these columns. I really only want 1, 2, 9, and 8 in that order. You will be doing the same thing down here for this data set by getting rid of pH minimum. So you'll want 1, 2, 3, 4, and then 6 to the end. So before you can go any further, please get rid of that pH-min column. I left that in there, and I did not put the code in now that I'm remembering on purpose. I wanted to get rid of it. And great. I actually did. I did include it. Oh, man. So forget everything I said about not putting it in, because I wanted you to look it up and do it yourself. Here's the code right there. This is putting the PA in front, then putting in the PA up front, and then the coordinates, and then the predictors. So there you go. You may follow this code. Okay. Model setup. Before we hit model setup, do DAT1, NA omit DAT1, meaning if there's any null values, we want to get rid of them. Okay. So the model to set up can be done within this code provided. So mod formula. It's going to be a function which requires your data, the R column, and the P column. So N column is the number of columns for the DAT. The response variable is going to be the column names for the DAT rows. So for the rows within the columns of the data. The predictor is going to be the column names for the all of the names that are past the coordinates in the PA column within this data set. And the mod formula will be as formula paste response, putting in this value which is required, based upon the predictor to the response variables within a generalized linear model, and we are pasting the predictors, and then everything in between will be added with the plus sign. So that was just a lot to explain in a terrible 20-second sentence. But you can just pretty much copy this code. This is just the code that sets up how we are doing this model. So for names, spec-trained data, now this is our new data set. We have PA, we have elevation, slope, aspect, p-average. Remember, we got rid of pH-min, and there are the rest of our predictors. So we also got rid of our latitude and longitude columns. We have just PA, and then it goes directly into our predictors. And this is all, these are all the values within this data set right here. Okay. So this is how you will then set up the formula to be used. Okay. So mod1l.lr, so model1.logistic regression. And so there's a function called GLM that will require as factor for our PA. So it is, our PA is either 0 or 1. It's not numeric. They are two different classes now, two different factors, 0 or 1. And based on that, this squiggly, what is it called? Oh, I always forget. Based upon that, we're going to look at the occurrences and how they interact with elevation plus slope plus aspect plus pH average plus the rest of our data. Then you will also put in family equals binomial, as this is a binomial between 0 and 1, and the data is equal to dat1, which would be the extracted variables. So that is what you can pretty much copy here. You won't need to change anything. You will just need to have previously changed your species, and this will be the same exact function because you're using the same exact variables. So now that we have run this generalized linear model, you can have a summary which shows you the formula which you provided. It shows you your residuals and your coefficients, your estimates, your standard error, the z-value. We're not going to be covering this. And honestly, you don't need to dive too deep into this at all. For this purpose and for this class, we want to look at just a few modeling metrics to understand how the model performed, and then how to spatially visualize your model. So just know that this is the output production of the generalized linear model. We're looking at modular fit. Not modular, that's not the right word. Model fit. Modular. And understanding thresholds. So kind of like what we did earlier, thresholds of species occurrence. Each modeling algorithm will produce a threshold, and you can just copy this code and run through it if you'd like to. I'm not going to cover it here. You can Google this information and see what's going on. And if you'd like to do more of this for your final project, sure, go ahead. But since we're behind, which is my fault, I don't want to keep you guessing on a lot of this stuff. I want you to move forward, be able to produce the output, and not hate your life. So we're creating two different model predictions. We're going to be looking at those values and how the models have predicted and how they're working. We're creating a fusion matrix, which is right here. It has to do with taking samples and understanding, taking X number of samples and withholding them from the algorithm. We're also going to be doing what's called within the thresholds of max kappa, and so max kappa will produce a modeling output between 0 and 1 for this study. So essentially, if you're using several different algorithms like random forest and maxent and generalized linear models and generalized additive models, their outputs are not standardized across those four outputs. You have to do a threshold based on max kappa, which is 0 to 1 probability. So we're assigning max kappa here. It's very important if you're doing more than one species distribution modeling to make sure that they are standardized to max kappa or to whatever standardization you prefer. Right here is very important for our modeling metrics is we are looking at the accuracy of the presences and absences and generating what is called sensitivity, specificity, AUC, and TSS. AUC stands for area under the curve of the receiving operator, and TSS stands for the true skill statistic. So following this code here, we are producing these. Presence, absence, accuracy function will generate the sensitivity, specificity, and AUC. The true skill statistic is derived from sensitivity plus specificity minus 1. And this mod 2 ACC will have all of these metrics, and we want metric 1, 4 through 5, and 7 through 8. 1 would be the model name, 2 would be sensitivity, or 4 would be sensitivity, 5 would be specificity, 7 and 8 are AUC and TSS. Now, sensitivity is essentially how well the model predicts a high suitability or presence. Specificity is the opposite, how well it produces absence or low suitability. AUC, the area under the curve, gives an idea of how well the model is performing based on the information we have provided, or is it just straight up guessing. And the true skill statistic is the difference between the sensitivity and specificity. So it's sensitivity plus specificity minus 1. Right? So the higher the value all of these are, ideally you have a better model. But AUC is by far the most used in model accuracy, and if it is above 0.75, you generally have a good model. Can range widely. One thing to note is that while you may produce a model with the AUC like 0.9 and one with 0.85, in reality, you cannot determine if one is better than the other based upon, truth of where these plants are located and where is real habitat suitability. But when you're modeling, you always want to have a higher AUC. So the AUC.ROC, so area under the curve of the receiving operator, plot, this is how we see it. So it's 0.857, so 0.86. We can see the line here, and then this is the straight line that goes from 0 to 1, so which would be, an AUC of 0.5. So the closer the AUC, this red line, is to the 0.5 little gray line here, essentially, for really lack of terms, it's bad. And that means that the model is just guessing and that it is not providing a true, estimate of, suitability based upon the algorithm. It doesn't know what it's doing. It didn't predict well. The higher this goes straight up to 1 and over, that means that the model predicted, much, much better. So 0.86 is fair and, pretty good. So based upon what we have created in our models thus far, it's a pretty good model. So here's a couple of trainings. So the cross-validation accuracies. You can cross-validate your models between two different models, as this is one, and we can cross-validate it to produce another confusion matrix and more accuracies assessments. And so we can do it once more and measure between model 1 and model 2 to see if there is any difference. For example, these are two different models. They produce different sensitivity, specificity, and AUC and TSS. And we can also plot their AUCs right here. So we can see how one worked. So 0.86, 0.85. One model did better in terms of predicting 0.86 to 0.85, but the difference is so small, and they don't measure reality anyway that, honestly, if you went with either one, they're fine. So you can fill in this code here, and this will produce these model outputs. Third one, but what you will need to produce for your homework is just at least one of these, the model prediction metrics, so sensitivity, specificity, AUC, TSS, for at least one model, right? Sweet, so that's where you cross-validate. Fill those in there. If you want to know more, you can feel free to Google that. And here is, once again, an RUC of the third. So there are ways to model, to examine how models compare with other models based upon cross-validation. But now that we have created the models, let's visualize them, which is by far the most exciting and probably pertinent point of this entire thing we're doing here. As many of you have no desire to dive further into species distribution modeling, some of you may. You really just want to produce something cool visually. And honestly, it's a real technique. It's a real modeling algorithm. And we'll be able to produce species distribution models using a generalized linear model. Okay, so spp.r or preds.preds.r, we're turning our predictors back into rasters from spat rasters. The generalized linear model prefers rasters. So we change our predictors, and I call this mod fprob.lr.1. You can call this whatever you want, prob1. We're using the predict function. So the predict function gathers our predictor variables, so all of those rasters. It uses our modeling outputs, right, based upon our trained data. And we can assign it a name. We can assign it a type, which is response, which will be the prediction. The function is predict, an index of 2, and to overwrite. In case you are saving this, this will produce a probability output. So from 0 to 100%, how probable a habitat suitability is. We will also be creating a classified version of the modeling output based upon a threshold classification that can be found in the code right here, based upon our max kappa, optimal thresholds, mod cut. So this is our cut threshold. So based upon max kappa and that threshold, everything above that value is going to be suitable. And this is just based upon modeling thresholds. We could also do a classified model probability, which would just be taking this 0 to 100 and putting it into 5 classes. So it will be 0 if it is less than 0.2, 1, 0.2 to 0.4, 2, you know what I'm saying. Actually, it's 0 to 0.2 would be 1, 0.2 to 0.4 would be 2. So it's just a way to break up the probability from 0 to 100 into 5 classes, 10 classes, whatever you'd like to choose. So let's plot them. So we have a plot where you're putting in our mod fprob.lr.1, which would be our probability. I'm calling it probability model. Right here we have 0 to the highest value possible within this model probability. And you can see here, it's looking pretty cool. It looks like the elevation. But you can see how, very low suitability to no suitability in the plains, and very high suitability in the mountains where you would find blue spruce. But it's not just choosing an elevation, it's actually using a generalized linear model based upon all of the predictor variables we include, and based on the presence and absence, it is showing us where highly suitable areas are for blue spruce. So this could be very useful in conservation planning, could be very useful in understanding what species might use blue spruce. It could be very useful in understanding future climatic change and how it affects species. And it kind of gives us an idea of where they occur in forested environments. So the Southern Rockies are highly forested throughout the entire thing, but blue spruce doesn't occur in all of them. So you can model this species and another tree species and get a good idea of community structure. And from there you could infer competition, things like that. You could look at disturbance, insects, fire, so on. The amount of studies you could use with species distribution modeling is endless. And if you followed along doing this, you have created a probability model for either your species or the Colorado blue spruce. Very cool. So based upon the threshold that we have created, this was our threshold code right here, right? Based upon mod cut, which is just a function on optimal thresholds for max kappa, saying that everything that is in green is going to be suitable areas for this species. This is kind of the value of the darker the green, the more higher the suitable, and this is just based on a threshold. Everything above this could be. And then we have bin probability, which is taking the probability that we see up here and just kind of structuring it into classes. It looks very similar, but can be helpful if you wanted to define it into more classes. And it has its uses. So like, take a look close here, take a look close there. you can see how that could be helpful. And you can look further into that and see how it might not be helpful. That was kind of like baptism by fire, drinking from the fire hose here for species distribution modeling. So in less than an hour, you have now the code to generate a species distribution model. Briefly explain what they do, what they don't do, and produce a spatial output. So it might take longer than an hour to code all this, but that's everything you would need to produce a generalized linear model. And so if we went further in depth, there might be more understanding on what some of these functions do. Gotten close to the end of the semester. I have delayed doing this by being out of town, submitting my thesis and defending next week, and so there's just a lot going on, and I know you all have a lot going on, and I don't want you to waste your time. So please follow along with this assignment and then gather your own species data and produce a species distribution model. And so if you're into species distribution modeling, we can talk further. I have a lot of resources for you to use, and if you'd like to use this for a final by examining a different area, I also have predictor variables for the either the world, North America, or the Western 11 states of North America, or of the United States. And so, based on what you're interested in, I have those predictor variables for you, so you're not spending all your time in life looking for those, as I did for the past 2 years. So, good luck. Let me know if you have any questions. I'll do my best to respond. I've been pretty bad at that, but with your patience, we'll get through it. And if you complete all your assignments, you can be confident you will pass this class with flying colors. See, it all works out in the end. So, all right, good luck.